Good evening, all. Am I audible to you?
Good evening, ma'am. Yes, ma'am.
Yeah, is my screen visible? My presentation
Yes,
slide?
ma'am.
Okay. Okay. So, good evening, everyone. I am Ananya Chatterjee. I'm a PhD student in IISc Bangalore. I'm one of three TAs of the course Introduction to Cell Biology, Cell Components, Organization, and Processes. So already I have covered week 1 and week 2 questionnaires and discussed some basic understanding and the uh, syllabus covered in those lectures. Today I will be going to discuss some questions from week 3 and also will cover some topics which were covered in the week 3 contents. So as you have already seen that week 3, week 4 and week 5 contents are live, so you can go through those lecture videos and please get back to us if you have any doubts or so. We will try to uh, answer about your doubts and we'll get back to you. So this course is taught by Professor Shikha Lalodaya from IISc, and she's a professor from Department of Biochemistry. So I'll be starting discussing about the week 3 assignment which was given to you, and yes, so let's start. So this is the first question, was that DNA replication must occur accurately. So I have to So the question is, DNA replication must occur accurately. Options are: to minimize the possibility of spontaneous mutation, to reproduce and maintain the correct genome organization, to ensure the duplication of all pairs of the chromosome only once per cycle. So by common sense we can understand that when cell divides, DNA also replicates, so that in the new cell or in the sister cells another set of DNA can go. So but while doing so, the cell need to while doing so the cell need to consider that if need to introspect that if any spontaneous mutation is happening while replication, it may happen. So that's why DNA replication must occur accurately. Different enzymes should check that whether there is any uh, there is any gap is there is any gap or any formation of mutation is happening in the DNA, and will do accordingly. So the first option is definitely correct, to reproduce and maintain the correct genome organization. This is also correct, to ensure the duplication of all pairs of the chromosome only once per cell cycle. So while we will duplicate the chromosome, we should have equal amount of chromosomes in each sister cells. Okay, so we have to, or the cell has to ensure that the duplication of all parts of the chromosome only occurs once per cell cycle. So we can get that all of the above will be correct. Then the next question was, pick the correct statements about the molecular structure of DNA. So we discussed about the molecular structure of DNA in previous class. The first option is the two strands in a DNA double helix run parallel to each other. A binds to T, C binds to G. The base pairs interact with each other by van der Waals interaction. The four bases present in DNA are AUGC. So you can see the A option is not correct. We know that the two strands of DNA runs in anti-parallel to each other, 5' to 3' and 3' to 5'. So the first option is incorrect. Second option is correct. A binds to T and G binds to C. Third option is also incorrect because it will be hydrogen bonds. So the fourth option, the four bases present in DNA are AUGC, but no, the four bases are ATGC. So this is also incorrect. So answer will be A binds to T. Now the third question was, pick the incorrect statement about DNA replication. First option is DNA synthesis occurs by extending the 3' end of the primer. DNA polymerase catalyzes the formation of phosphodiester bond. NTPs are utilized as substrate for addition of new nucleotides. During phosphodiester bond formation, pyrophosphate is released. So you can see that the wrong statement is NTP because DNA is made up of DNTPs, not NTPs, okay? And we know that primer will add and that will be extended towards 3' end. Also, DNA polymerases catalyzes phosphodiester bond, this is correct. And during phosphodiester bond, pyrophosphate is released, that is also correct. Now we'll talk about the next question. Yeast replication origins are called. So this is, you have to know that yeast replication origins are called ARC or autonomously replicating sequence. So what is ARC? So this is a yeast chromosome. So you can see so this is the yeast chromosome, where you can see that this is a chromosomal DNA insert. This is a selectable marker gene. Then it will be amplified and library of different inserts will be there in library. So you can see that what is autonomously replicating sequence. An autonomously replicating sequence contains the origin of replication in which in the yeast genome. So, and also for additional information, it contains 4 regions. So it contains 4 regions A, B1, B2, B3 named in order of their effect on plasmid stability. So the answer, which is the correct answer? ARS is the correct answer. Now the next question was, the regulatory point in animal cells which is crucial for controlling the entry into S phase of cell cycle is termed as restriction point. As we know that normal cells, what happens, they can withdraw growth factors, okay, to prevent the onset of S phase. So if they need to stop or they need to control the entry of the cell into S phase, they need to withdraw some growth factors which may be essential for S phase to prevent their onset to S phase. So the point at G1 at which commitment occurs and the cell no longer requires growth factors to complete the cell cycle has been termed as restriction point. Now, what was the second question? Pick out the correct role played by RB by retinoblastoma in cell cycle. So, retinoblastoma protein is a tumor suppressor, okay, which plays a important role in the negative control of the cell cycle and tumor progression. So it has been shown major G1 checkpoint, and it also block S phase entry and cell growth, hence cell division. So what are the options? Retinoblastoma acts as a transcription factor activator. E2F retinoblastoma bound complex activates E2F. Retinoblastoma is phosphorylated by G2M CDK. Active RB inhibits the entry of cells into S phase. So the correct role, we can see that active RB inhibits the cells into S phase from G phase. The phosphorylated retinoblastoma protein represses gene transcription required for transition from G1 to S phase by directly binding to the transactivation domain of E2F and by binding to the promoter of genes as a complex with E2F. Okay, so what happens when the PRB protein represses gene transcription and required for they represent those the transcription of those genes which are required for transition from G1 to S phase. So how they do, they directly bind with the transactivation domain of E2F and by binding to the promoter of those genes. So PRB represses transcription also by remodeling chromatin structure through the interaction with proteins such as HBRM, BRG1 and so on, which are involved in nucleosome modeling, histone acetylation and deacetylation, methylation respectively. Loss of PRB function may induce cell cycle deregulation and so lead to malignant phenotype. So then we can see that, so this is the diagram depicts the same thing, that retinoblastoma and cell cycle machinery. Retinoblastoma and RBP represents the unphosphorylated and phosphorylated respectively. So what happens in G0 and early G1 phase, retinoblastoma physically associates with E2F factors. And what they do, they blocks their transactivation domain. So then what happens, they eventually will block the transcription. And in late G1, retinoblastoma is phosphorylated and releases E2F so that E2F can function. And also what happens, it allows the expression of genes that encode products necessary for S phase progression. Now the next question is, the pre-RC complex consists of, so what is RC? Pre-replication complex pre-replication complex consists of ORC, CDC6, ORC and 2 copies of MCM complex, CDT1 and MCM complex. So you have to understand that this is pre-RC complex, okay. So the answer is ORC, origin of replication complex, and 2 copies of MCM complex. This is the right answer. Next question was activation of MCM helicase subunits is carried out by DDK. This is a kinase, okay, which modifies MCM helicase. We always know that what kinase do, kinase phosphorylates. So what it does, it will phosphorylate only. Next question, the semi-conservative model of DNA replication was experimentally demonstrated by Michelson and Starr, 1958. During DNA replication, in which direction does the DNA polymerase 3 synthesize the new DNA strand? 5' to 3', 3' to 5', 3' to 3', and 5' to 5. Obviously, 5' to 3'. Now this is week 3 assignment previous year. The question was, ubiquitin is attached to which position of lysine amino acid? So ubiquitin will be attached at gamma position. Okay, it will bind to alpha, beta, gamma, delta, epsilon. So it will bind to epsilon position, not delta. It will bind to epsilon position. Which of the following factors is not required which of the following factors is not required for formation of pre-replicative complex on origins? So the answer is phosphorylated MCM proteins. So the next question is, which of the following is present in proteins of the ubiquitin-like protein family? So we will be discussing about ubiquitin-like protein family. So you can see there that which of the following is present? 5-stranded beta sheet, alpha helix, short 310 helix, all are present in the ubiquitin-like protein family. So what are ubiquitin-like protein family? The family of small proteins related to ubiquitin called ubiquitin-like proteins share similarity in 3D structure. So they have a characteristic ubiquitin fold or beta grass fold. It consists of a 5-stranded beta sheet, an alpha helix and a short 310 helix. The mature form of ubiquitin-like protein or ubiquitin-like polypeptide family ends with a signature diglycine sequence that is often exposed by processing of a ubiquitin precursor polypeptide. The C-terminal residues of the ubiquitin-like family is a glycine, and the carboxyl group of this glycine is a site of attachment to substance. Lysine side chains are the most common targets within substrate proteins, resulting in an amide bond between ubiquitin-like protein and substrate. Now we'll go to the next question. Pick out the odd one out from the given option: ring domain, Hecti domain, U-box, destruction box. So to understand that, we need to know that what are these proteins. So ubiquitin and U-box, Hecti box and ring-like domain all are part of E3 ligase. Okay, so we know that during ubiquitin annihilation we have E1, E2 and E3 ligases. So in E3 ligases there are four types. Two main types E3 ubiquitin ligases are ring and Hecti, and there are also U-box and PhD finger. So what are ring? What are the constituent of ring? It has a subunit or domain with a ring, really interesting new gene motif, which coordinates a pair of zinc ions. Function partly as adapters. It binds with ubiquitin thioester linked E2 and substrate protein simultaneously and position the substrate lysine nucleophile in close proximity to the reactive E2 UBL thioester bond, facilitating transfer of the ubiquitin-like protein. And also it triggers a conformational change in the bound E2, stimulating ubiquitin-like protein release from the E2 cysteine and its transfer. And there are another ligase, it is Hecti, This is also mechanically similar with the rin E3 ligase. So it has a HEC domain. The full form of HEC is homologous to the E6AP carboxyl terminus. Ubiquitin is first transferred from E2 to an active site cysteine in the conserved HEC domain of E3. Now the next question is, which of the following has been implicated in the regulation of histone stability for controlling gene expression? So here the options are heterotypic ubiquitin chains, correct, branched ubiquitin chains, APCC, K1148 ubiquitin chains, and all of the above. So the answer will be all of the above. So this all will help in stimulating histone stability for controlling gene expression. The next question is, select the odd one out from the given options: ubiquitin, NET8, SUMO 1 and THIS. So these all ubiquitin, NET8, SUMO 1 are best characterized ubiquitin like protein. So this will come into one group, and this is a sulfur carrying protein, this will come in another group. So this ubiquitin like protein are involved in regulation of the activity and the stability of diverse components of various oncogenic and tumor suppressor pathways, and this deregulation of enzymes responsible for their conjugation and deconjugation has also been associated with tumorigenesis and cancer resistance to therapies. The ubiquitin like system therefore constitutes so it is considered as attractive target for developing novel anti-cancer therapeutic strategies. Now the next question was, in which phase of the cell cycle does licensing of replication origins occurs? Licensing of replication origin means whether the cell should proceed through replication or not. So this occurs at the end of M phase and G1 phase, that is just before S phase. So answer will be this. Which of the following statements is incorrect for S phase of cell cycle? Options are, S phase is also known as a synthesis phase of cell cycle. This is correct. The entry of a cell into the S phase is highly regulated. This is also correct. You have seen various checkpoints, various proteins which will be regulated, which will be activated, down regulated, so that the S phase is highly regulated. Once a cell passes through the G1 restriction point or start, it gets committed to go through the S phase. This is also correct. Removal of external stimuli inducing entry into S phase causes the S phase cells to go back. No, this is not the correct answer. Now the next question is, external growth factor induce the synthesis of D-type cyclin in mammalian cells. The cyclin is important for the cells to cross the restriction point, inactivation of ORC, inactivation E2F. So we know that cyclins are responsible for to cross the restriction point. Which is the catalytic subunit of 26S proteasome? So the catalytic subunit is you have to know that the catalytic subunit of 26S proteasome is 20S subunit. Now I will cover some theoretical portions from week 3. So what are germ cells and somatic cells? So we know the cells of a sexually reproducing animal or plant are of two types. One is germ cells and another one is somatic cells. So what does germ cell do? The germ cell transmits genetic information from parent to offspring, and the somatic cells form the body of the organisms. We have seen that germ cells must be protected against high rates of mutation to maintain So we have seen that the germ cells must be protected against high rates of mutation to maintain the species. However, the somatic cells of multicellular organism must also be protected from genetic change to properly maintain the organized structure of the body. Nucleotide changes in somatic cells can give rise to variant cells, some of which, through local natural selection, proliferate rapidly at the expense of the rest of the organism. In an extreme case, the result is the uncontrolled cell proliferation that we know as cancer, a disease, and this dates. This is the consequence if something go wrong with the proliferation of cells. So if we see two gametes when fused, it will form a zygote, and from that this red cells are germline cells, and within this some cells will be somatic cells. So this will eventually divide and form the body. So germline cells and somatic cells carry out fundamentally different functions. In sexually reproducing organisms, germline cells propagate genetic information into the next generation. And what does somatic cell do? They form the body of the organisms, which is necessary for the survival of germline cells, who do not themselves leave any progeny. Now we will come to DNA replication. So in the DNA double helix, we know that one strand acts as a template strand for its own duplication. So you can see this is a one strand called S strand, is a S prime strand. So this is a parent DNA double helix. So if DNA replicates, one strand will be acted as a template to generate another new strand, and so on. So because a nucleotide A will pair successfully only with T and G with C, each strand of DNA can serve as a template to specify the sequence of nucleotides in its complementary strand by DNA base pairing. In this way, the double helical DNA molecule can be copied precisely. So if the DNA one of the strands had ATGC like that sequence, and if the other strand is getting replicated, so nucleotides will come like that way only, so that against A, T nucleotide will come and bind by hydrogen, interact by hydrogen bonding. So this is how the precision will maintain. So here you can see that 3' end of strand and then 5' end of strand, and this is the backbone where those bases are projected away. The addition of deoxyribonucleotide to the 3' end. So when how DNA getting replicated 5' to 3'. So what happens in the 3'? The addition of DNA like deoxyribonucleotide happens to the 3' end of polynucleotide chain, is a fundamental reaction by which the DNA is synthesized. So the base pairing is happening between the incoming deoxyribonucleotide nucleoside triphosphate and the existing strand, and it is adding a new nucleotide, and for that is how the DNA strand is getting extended. So this experiment you have seen in the lecture. So this is a Michelson style experiment, which basically proves how the semi-conservative replication happens. So if there is nitrogen is 15 nitrogen is present, that is radioactive nitrogen is present inside DNA, and if you grow cells in a normal isotope of containing normal nitrogen media, then what will happen is that when DNA will replicate, they won't be able to get radioactive level nitrogen, so they will incorporate non-radioactive nitrogen, that is 14, and so on, and this is how it will grow. And eventually what will happen, that after some time there will be equal number of heavy DNA and light DNA, and so these are the experiment by, and after that how they could prove, they isolated from cells and mixed with cesium chloride solution, and then they did ultracentrifugation density gradient, and that's how heavy chain to light chain, that's how they got segregated in separate layers. So what is density gradient ultracentrifugation? So what happens here, under high centrifugal force, a solution of cesium chloride molecules will dissociate, and the heavy cesium plus atoms will be forced away from the center towards the outer end of the tube, but will at the same time diffuse back towards the top of the tube. Thus it forms a shallow density gradient, and DNA molecules placed in this gradient will migrate to the point where they have the same density as the gradient, the neutral buoyancy or the isopycnic point. The gradient is sufficient to separate types of DNA with slight differences in density due to differing G plus C content or physical form. So what happens in this experiment above, after centrifugation for 10 hours at 1 lakh rpm, two distinct bands corresponding to shared linear nuclear DNA above and circular mitochondrial DNA below are visible under ultraviolet light. The DNA has been mixed with the intercalating dilithium bromide, which enhances the density difference between the two forms and causes the DNA to fluorescence. The separate bands are collected by poking a hole. So this separate bands you can collect by poke a hole in the bottle and the bottom of the tube, and the intact mitochondrial DNA will be available for experiment. So as you have already seen in the lecture, DNA replication happens in ACE phase of the cell cycle after G1 phase. So this entry is highly regulated. A regulatory point in G1 is termed as restriction point. So it is a decision point at which the cell evaluates whether conditions are right to proceed to S. In yeast, external signals such as nutrient availability, cell size, the presence or absence of mating pheromone affect So, in yeast, external signals such as nutrient availability, cell size, and presence or absence of mating pheromone affect the outcome. In animal cells, external growth factors signal proliferation in presence of these external growth factors, and cells cross the restriction point. Passage through the restriction point is irreversible. Now the cells is committed to go through S and the other steps of cell cycle, even if the external stimuli are withdrawn. If appropriate conditions are not available in G1, the cell cycle progression stops at the restriction point. Cells can arrest and enter a quiescent state of the cell cycle called G0. So now we will be discussing what are the key regulators of cell cycle transition and progression. In yeast, passage through start CDK1 in association with G1 cyclin, cyclin 1, 2, and 3 are the regulators to passage through the start site, and progression through S. Progression through S, CDK1 with S cyclins, that is CLB5 and CLB6, entry into and progression through mitosis, Any question, Abhishek? Do you have any question, Abhishek Ahirwar? You have raised your hand. Okay, so if not, we will proceed. So, entry into and progression through mitosis are regulated by CDK1 with M cyclins and CLB1 to 4. So, you have to memorize these names. In animal cells, progression through restriction points CDK4, CDK6, and cyclin D. G1 to S phase will be regulated by CDK2 and cyclin cyclin E. Progression through S to G2 is regulated by CDK2 and cyclin 1. G2M is regulated by CDK1 cyclin A, and progression through M is regulated by CDK1 and cyclin B. So here you can see if there is any growth factors, and by this RAS, RAF, MEK and ERK pathway signaling, that will synthesis D-type cyclins, and it will eventually regulate CDK4 and 6 and cyclin D, and will help the cell to cross this restriction point. So you have seen this slide in the lecture also. So these are the mitosis stimulation of cell cycle entry, G1 CDK activation and commitment. So these are the growth factor that RTK, this is the RTK domain, tyrosine kinase domain, where this growth factor will bind, and then that RAS, RAF and MEK signaling pathway will be upregulated. That ERK will eventually bind with the gene transcription and will increase the transcription of essential genes, and hence cell crosses restriction point. Okay, so here also you can see that phosphorylation of retinoblastoma gene or retinoblastoma protein can hamper in upregulation and downregulation, like way that if retinoblastoma is not phosphorylated, it will bind with E2F, and it will stop the genes which are necessary for S phase. It will off, and if it is getting phosphorylated, then the G1 CDK will be from the G1 CDK we'll see G1 S CDK will be activated, and the cell will entry into S phase. The normal pathway will be intact. So these are the additional regulation of CDKs by cyclin inhibitory kinase in mammalian cells. So you have to memorize these names. So the inhibitor family 1, INK4 family, P15, 16, 18 and 19. So they regulate CDK or CDK cyclin complex, CDK4 and CDK6, and due to that G1 phase is getting inhibited. Another family is CIP or KIP family. They regulate CDK2 and cyclin E complex and CDK2 and cyclin A complex, and hence G1 and SG2 cell cycle phase are getting inhibited. So what happens in deregulation of cell cycle in cancer? Cyclin D is a critical target of growth factor signaling. So if there is defect in cyclin D regulation, result in loss of growth regulation of the entry into the cell cycle as seen in cancer. Example, mutation causing continued unregulated expression of cyclin D contribute to lymphomas and breast cancer. Mutations that inactivate INK CKIs that bind and inhibit CDK4 and CDK6 are also common in human cancers. The CDK4/6 cyclin D target retinoblastoma is frequently mutated in retinoblastoma and a variety of human cancers. RB is a tumor suppressor. It binds E2F and represses E2F mediated transcription of S-phase genes. Its inactivation may allow S-phase entry and progression similar to when it is phosphorylated by G1 CDK and dissociates from E2F. So when everything goes right, then cell prepare for S phase. That is also called licensing of ORIs or ORI sequence, origin of replication. The initiation of replication is divided into two steps that occur at different phases of cell cycle. So in late M and early G1, a pair of replicative helicases, also called MCMs, bind at origins, forming the pre-replicative complex. This is termed licensing of replication ORIs. Initiation in S phase can occur only at those origins that have pre-RC. In S phase, DNA helicases are activated, resulting in DNA unwinding and initiation of DNA synthesis. The helicase moves out of the ORIs along with the RFs. The origin site now lacks a bound helicase and cannot reinitiate replication until after it binds again in the next G1. Hence, origins can be activated only once per cell cycle. So, how this regulation of initiation of DNA replication happens? Step 1 will be licensing. It is late M and early G1. So a large multiprotein complex, the origin recognition complex or ORC, is bound to replication origin throughout the cell cycle. It serves as a landing pad for several additional proteins that regulate initiation of replication. CDC6 is present at low levels during most of the cell cycle. Its levels increase transiently in early G1. It binds to ORC at origins in early G1. CDC6 is required for the beginning of a complex composed of group of closely related proteins, MCF proteins. Another protein, CDT1, also associates with the helicase and helps in loading it. CDT1 helps loading MCM helicase to the ORC and CDC6, hence to the DNA. So ORC, CDT1 and CDC6 collectively help in loading collectively help in loading two copies of the inactive helicase around the DNA next to the origin. The resulting large complex formed at an origin is known as pre-replicative complex or pre-RC. So after this process, now the origin is licensed. So pre-replicative complex consists of ORC, MCM helicase in two copies. Now regulation of initiation of DNA replication. Step 2, what is initiation of replication? So in S phase, S-CDK stimulates assembly of several initiator proteins on the helicase to help in its activation. Another protein kinase, DDK, phosphorylates helicase subunits and hence activating it. The activated helicase unwind DNA. DNA polymerase and other replication proteins are recruited to ORIs and DNA replication starts. ORC is displaced but rebinds. So these are the initiator protein and S-CDK binds. Inactivation of ORC, CDC6 and CDT1. DDK comes and phosphorylates MCM. MCM helicase unwinds the DNA. And what happens? The DNA polymerase and other replication proteins are recruited to ORIs and DNA replication starts. So this is DNA polymerase and other proteins are recruited. So DNA replication is initiated only once per cell cycle by S-CDK. Concurrent with initiation of replication, SCDK inactivates the pre-RC components ORC and CDC6 and prevents reformation of new Concurrent with initiation of replication, SCDK inactivates the pre-RC components ORC and CDC6 and prevents reformation of new pre-replication complex till the end of mitosis. So it dissociates CDC6 from origins, and also it is degraded by SCF that recognizes phosphorylated CDC6, and eventually it ubiquitinates and then it will get degraded by proteasomes. ACDK also phosphorylates certain MCM proteins, which triggers their export from the nucleus, further ensuring that MCM protein complex cannot bind to origins. So this is how CDC6 and MCM cannot return and rebind to to reset an ORC containing origin for another round of DNA replication. So until MCDK has been inactivated later. So this is a very important question can be asked, or important question to understand that why DNA replication is initiated only once. So this is one reason. Another is the E3 ligase APCC is turned off in late G1. Its target geminin, and CDT1 inhibitor accumulates and inhibits free CDT1. In addition, CDT1 is at active forks associates with protein that stimulates its destruction. So this is how the proteins which are required for pre-replication complex formation, these are getting degraded. So hence pre-RC formation is prevented from S phase until mitosis, ensuring that ORI is fired only once in a cell cycle. So to summarize, limiting DNA replication to once per cell cycle, ORC binds ORIs throughout the cell cycle, serve as landing pads for different regulatory proteins, example CDC6. So levels increase in early G1 binds ORC. CDC6 at ORI results in binding MCM protein and hence form pre-replication complex. Activation of SCDK initiate ORI firing assembly of DNA polymerase and other replication proteins, activates MCM rings to migrate along DNA strands as helicals, and prevents re-replication. Phosphorylates ORC, CDC6 gets dissociated from ORI and disassembly of pre-RC happens. CDC6 phosphate hence ubiquitination of SCF and degraded by proteasome. MCM also exported from nucleus can't come again into nucleus. Thus CDC6 and MCM cannot reassemble. So now SCDK maintains high activity during G2 and early MCDK also phosphorylates CDC6 and MCM prevent re-replication. At the end of mitosis, CDK activity reduces, dephosphorylation of CDK6 and MCM, pre-RC assembly at ORI, readies DNA for another round of replication. Now we will understand DNA replication step by step. So what happens, this is a DNA strand 3' to 5' and 5' to 3'. So this is primer strand. Now one triphosphate will come and bind, and one pyrophosphate will be released, phosphodiester bond will be formed, and DNA replication will continue 5' to 3' direction. So this is a DNA polymerase which holds the DNA, the temple strand as well as primer strand, and then DNA new strand will be generated and will go. So this is how it ensures the correct positioning of incoming deoxyribonucleoside triphosphate. So this is leading strand and these are lagging strands. So it forms like 5' to 3' as it some amount of DNA will unbound and then will replicate the lagging strand. So it's also called Okazaki fragments. Okay, so these are most recently synthesized DNA. So this is at first this will synthesize, then some amount will again unbound, and then this will synthesize, and like that. So this is a replication fork structure. So here you can see replication fork with newly synthesized DNA in red, and arrows indicating 5' to 3' direction of DNA synthesis. Because both daughter DNA strands are polymerized in the 5' to 3' direction, the DNA synthesized on the lagging strand must be made initially as a series of short DNA molecules called Okazaki fragments, named after the scientist who discovered it, and ride the same fork for a short time later. On the lagging strand, the Okazaki fragments are synthesized sequentially, with those nearest the fork being the most recently made. Exonucleotide proofreading by DNA polymerase during DNA replication. So this is primer strand and template strand. So transiently see what happens, that we need T against A, okay, but if C transiently base pairs with A and is incorporated by DNA polymerase into the primer strand, what happens, this will eventually unpaired 3', so the 3' OH will be unpaired. End of primer blocks further elongation of primer strand by DNA polymerase. 3' to 5' exonuclease activity attached to DNA polymerase shews back to create a base pair 3' OH. It will shew it and will create a base pair 3' OH on the primer strand. Then DNA polymerase again resume by adding thymine in the process, and will base pair with a 3' OH. Editing by DNA polymerase. DNA polymerase complexed with DNA template in the polymerizing mode and the editing mode. So the catalytic site for the exonucleotide and polymerization reactions are indicated here. In the editing mode, the newly synthesized DNA transiently unpairs from the template and enters into the editing site, where the most recently added nucleotide is catalytically removed. And while the polymerization will go on, this will go by polymerization site, and where the DNA replication will be continued. So this is how RNA primer synthesis occurs. So here you can see a view this reaction catalyzed by DNA primase. So this what does this enzyme do? It synthesizes the short RNA primers made on the lagging strand using DNA as a template. So unlike DNA polymerase, this enzyme can start a new polynucleotide chain by joining two nucleotide triphosphate together. The primase synthesizes a short polynucleotide in the 5' to 3' direction and then stops, making the 3' end of the primer available for the DNA polymerase. The synthesis of one of many DNA fragments on the lagging strand. So this is lagging strand template 3' to 5'. So you can see RNA primer, and this is a new RNA primer synthesis by DNA primase. So the thing will be synthesized like this, 5' to 3' direction, so that the new nucleotide will bound with the 3' OH. So DNA polymerase adds two new DNA primer to start new Okazaki fragments. DNA polymerase finishes DNA fragment, old RNA primer erased and replaced by DNA. Sealing by DNA ligase joins new Okazaki fragment to the growing chain. So in eukaryotes, the RNA primers are made at intervals spaced by about 200 nucleotides on the lagging strand, and each RNA primer is approximately 10 nucleotide long. The primer is erased by a special DNA repair enzyme, RNase H, that recognizes an RNA strand in an RNA DNA helix and fragments, and it leaves gaps that are filled in the DNA, filled by the DNA polymerase and DNA ligase. So, how the reaction catalyzed by DNA ligase? This enzyme seals a broken. So, what does this DNA ligase do? This enzyme seals a broken phosphodiester bond, as shown. DNA ligase uses a molecule of ATP to activate the 5' end at the nick before forming the new bond. In this way, the energetically unfavorable nick sealing reaction is driven by being coupled with the energetically favorable process of ATP hydrolysis. So you can see the 5' phosphate and 3' OH, you need to make the bond, and while doing so, one ADP will be used and will form ADP, and the phosphate will then bind with the OH, and eventually another phosphate will release and AMP will be released. So, some special proteins that help to open up the DNA double helix in front of the replication fork. For DNA synthesis to proceed, the DNA double helix must be opened up ahead of the replication fork so that the incoming deoxyribonucleoside triphosphate can form base pairs with the template strands. However, we know that DNA double helix is very stable under physiological conditions. The base pairs are locked in place so strongly that it requires temperatures approaching that of boiling water to separate the two strands in a test tube. For this reason, two additional types of replication proteins, DNA helicase and single strand DNA binding proteins, are needed to open the double helix and provide the appropriate single strand DNA templates for the DNA polymerase to copy. DNA helices were first isolated as proteins that hydrolyze ATP when they are bound to single strands of DNA, and the hydrolysis of ATP can change the shape of a protein molecule in a cyclical manner that allows the protein to perform mechanical work. So, DNA helicals will bind and then will help in unwinding the DNA, or slightly opens up the one strand from another. So the two strands of DNA have opposite polarities, and in principle a helicase could unwind the DNA double helix by moving the 5' to 3' along one strand, or in the 3' to 5' along the other. In fact, both types of DNA helicase exist. In the best understood replication system in bacteria, a helicase moving 5' to 3' along the lagging strand template appears to have predominant role. Now we'll discuss about single strand DNA binding protein. So single strand DNA binding proteins, also called helix destabilizing proteins, bind tightly and cooperatively to exposed single strand DNA without covering the bases, which therefore remain available as template. These proteins are unavailable to open a long DNA helix directly, but they aid helicases by stabilizing the unwound single strand conformation. In addition, through cooperative binding, they coat and straighten out the regions of single strand DNA which occur routinely in the lagging strand template, thereby preventing the formation of the short hairpin helices that readily form in single strand DNA. If not removed, these hairpin helices can impede the DNA synthesis catalyzed by DNA polymerase. So these are single stranded binding monomers. It will bind like that so that it stables the single chain of DNA. The effect of SSB protein on the structure of single strand DNA, because each protein molecule prefers to bind next to previously bound molecule. Long rows of this protein forms on a DNA single strand. and this cooperative binding straightens out the single stranded DNA and facilitates the DNA polymerization process. And the hairpin helices shown here, single strand DNA results from a chance matching of short regions of complementary nucleotide sequence. They are similar to the short helices that typically form in RNA. Now we will discuss about a sliding ring holds a moving DNA polymerase onto the DNA. So this is a clamp loader and this is a sliding clamp. So clamp loader will load the sliding clamp and eventually the DNA will pass by, and what happens, the DNA polymerase will be loaded on the clamp loader and it will hold the DNA polymerase. So what is the story behind it? On their own, most DNA polymerase molecules will synthesize only a short string of nucleotides before falling off the DNA template. So DNA polymerase will, if DNA polymerase alone will bind with DNA, it may fall off, and it will make before falling off, it will make a short string of nucleotides. The tendency to dissociate quickly from a DNA molecule allows a DNA polymerase molecule that has just finished synthesizing one Okazaki fragment on the lagging strand to be recycled quickly, so as to begin the synthesis of the next Okazaki fragment of the same strand. This rapid dissociation, however, would make it difficult for the polymerase to synthesize the long DNA strands produced at a replication fork, if not for an accessory protein called pCNA in eukaryotes that function as a regulated sliding clamp. This clamp keeps the polymerase firmly on the DNA when it is moving, but releases it as soon as the polymerase runs into a double strand region of DNA. Now the question is, how can a sliding clamp prevent the polymerase from dissociating without at the same time impeding the polymerase rapid movement along the DNA molecule? The three-dimensional structure of the clamp protein determined by X-ray diffraction revealed that to be a large ring around the DNA double helix. One face of the ring binds to the back of the DNA polymerase and the whole ring slides freely along the DNA as the polymerase moves. The assembly of the clamp around the DNA requires ATP hydrolysis by a special protein complex clamp loader. On the leading strand, the moving DNA polymerase is tightly bound to the clamp and the two remain associated for a very long time. The DNA polymerase on the lagging strand also makes use of the clamp, but each time the polymerase reaches the 5' end of the proceeding Okazaki fragment, the polymerase releases itself from the clamp and dissociates from the template. So ATP binding to clamp loader and open sliding clamp. So at first the sliding clamp is closed. When ATP binds with the clamp loader, it will open it. DNA engaged in clamp, and again ATP hydrolysis lock it. And then if DNA polymerase binds, the clamp loader will be recycled back, and then DNA polymerase the sliding clamp will hold and slide the DNA polymerase through the DNA. So at the site of replication fork, different other proteins are also comes and then eventually form a replication machine. So what are the proteins at the replication fork? You can see these are single strand DNA binding protein we discussed to stable that single stranded DNA. Then DNA primase will form a short primer, sliding clamp or and clamp loader, DNA polymerase, DNA helicase, and so on. So So these are the protein complex formed in the replication fork region. Now what happens if there is some error happens during DNA replication? So this is a strand directed mismatch repair system removes replication errors that escape from the replication machine. So if an error is there in newly made strand, what happens? Two proteins bind with it, mutase and mutel, and they they scan DNA for any defect, and ultimately it cuts one strand and then remove that strand, and again the repair of DNA synthesis happens. So there are two proteins in both bacteria and eukaryote. Mutase binds specifically to a mismatched base pair, while mutel scans the nearby DNA for a nick. So when mutel finds a nick, it triggers the degradation of nicked strand all the way back through the mismatch. Because nicks are largely confined to newly replicated strands in eukaryotes, replication errors are selectively removed. In bacteria, additional protein in the complex mut-H nicks unmethylated and therefore newly replicated GATC sequence, thereby beginning the process here, and then eventually repair or the DNA synthesis will be started. So, what is the role of DNA topoisomerase during replication? So, they prevent DNA tangling during replication. How do they do that? So, when a replication fork moves along double strand DNA, it may create a winding problem. So the two parental strands which are wound around each other must be unwound and separated for replication to occur. So for every nucleotide pairs replicated at the fork, one complete turn of the parental double helix must be unwound. In principle, this unwinding can be achieved by rapidly rotating the entire chromosome ahead of a moving fork. However, this is energetically highly unfavorable, particularly for long chromosomes. Instead, DNA in front of a replication fork becomes overwound. The overwinding in turn is continually relieved by proteins known as DNA topoisomerase. So if there is a long strand of DNA and it's not able to move properly, what happens? The DNA cannot rapidly rotate and torsional states will build up. Here also, if you see, if you just try to open up the chain, it may become overwinding. So this is how topoisomerase comes into play. What there are two kinds of topoisomerase, 1 and 2. So what does topoisomerase 1 do? You can see this is one end of DNA double helix cannot rotate relative to the other end. So type on DNA topoisomerase with tyrosine. So it has a tyrosine amino acid at its active site. It comes and it will bind with one bases. So DNA. So it will bind with the backbone of DNA. Okay. So it covalently attaches with DNA phosphate. So with OH, it has a OH group. So it will bind with phosphate of DNA backbone, and thereby breaking the phosphodiester bond linkage in one DNA strand. Then there is a OH group, you can see. So eventually the two ends of the DNA double helix can now rotate. So this can be rotated and the torsional stress will be gone. The original phosphodiester bond energy is stored in the phosphotyrosine linkage, making the reaction reversible. And spontaneous reformation of the phosphodiester bond regenerates both the DNA helix and the DNA topoisomerase. Now the DNA helix passing reaction catalyzed by DNA topoisomerase 2. Unlike type 1 topoisomerase, type 2 enzymes hydrolyze ATP, which is needed to release and reset the enzyme after each cycle. Type 2 topoisomerase are largely confined to proliferating cells in eukaryotes. Partly for that reason, they have been effective targets for anti-cancer drugs. Some of these drugs inhibit topoisomerase 2 at the third step. So when two circular DNA double helices that are interlocked, topoisomerase will come and will break phosphodiester bond in two strands, in two circular DNA double helices only. So they recognizes the entanglement and makes a reversible covalent attachment to the two opposite strands and will pass one strand over another. Uh, then the gate shuts, releasing the red helix. So what happens? The reversal of the covalent attachment of the topoisomerase restores an intact orange double helix. So you can see these two strands, helices were entangled, and then it will recognize, and then the entanglement makes reversible covalent attachment to the two opposite strands of one of the double helices, creating a double strand break and forming a protein gate. The topoisomerase gate opens to let the second DNA helix pass. So this is DNA replication, prokaryotic DNA replication during cell division. So you can see this is the parent cell DNA and origin of replication, where we have already discussed different proteins come into play. They form a replication fork and so on, and then eventually the replication ends. So in prokaryote, the chromosomes is circular. Replication initiates at an origin or initiator sequence. Origins binds Origins bind a sequence specific binding protein, the initiator protein. Origins also have an easily unwound region, AT reach. Replication initiates with the help of initiator protein and primosome. So here DNA helicase and primase helps in replication initiation. DNA polymerase synthesizes leading as well as lagging strand. Replication proceeds bidirectionally until they collide at the end and the primosome is disassembled. So some key requirements for the DNA replication. Initiator, also called DNA A, binds with origin DNA and melts the AT rich region. Helicase, it unwinds, this is also called DNA B, unwinds DNA at the replication fork to generate single strand DNA template strands. Primase, DNA G, synthesizes the RNA primer for DNA synthesis. DNA Pol 3 hollow enzyme. Clamp loader, it loads sliding clamp on a primer template duplex. Tau protein, it connects clamp loader to DNA Pol 3 core, also connects and stimulates helicase. DNA Pol 3 core does DNA synthesis. Sliding clamp DNA polymerase 3 core and enhances its processability. Single strand binding protein prevents annealing of single strand DNA template strands. DNA Pol 1, primer removal, DNA repair, ligation of Okazaki fragments. Topoisomerase, we just discussed, prevent DNA tangling. So we have already covered these things. The protein which initiate DNA replication. This is replication origin, parental DNA. Binding of initiator proteins. So these are initiator proteins and DNA helicase. It will unwind and then the primase will bind, will make primer. DNA polymerase will begin leading strand and lagging strand, and two replication forks moving in opposite direction. So you can see this is fully methylated origin and these are hemi-methylated origin. So for methylation of E. coli replication origin creates a refractory period for DNA initiation. So DNA methylation occurs at GATC sequence. Any question, Thirupu? So where does DNA is methylated? DNA methylation happens at GATC sequence. So this kind of sequence around 11s are found. So in the hemi-methylated state, what happens? The origin of replication is bound by an inhibitor protein, also called SEC-A, which blocks the ability of the initiator proteins to unwind that origin DNA. Eventually, the hemi-methylated origins become fully methylated by a DNA methylase enzyme. SEK then gets dissociated. A single enzyme called DAM methylase is responsible for methylating all E. coli GATC sequence. A lag in methylation after the replication of GATC sequence is also used by the E. coli mismatch proofreading system to distinguish the newly synthesized DNA strand from the parental DNA strand. Now you can see there is a formation of nucleosome behind the replication fork. So besides the replication of besides forming a new strand, there should be some reformation in the nucleosomes also. So in this diagram, if you see, you can see these are parental H3 H4 tetramers are distributed at random to the daughter DNA molecules with roughly equal numbers inherited by each daughter. In contrast, H2 and H2B dimer are released from the DNA as the replication fork passes. This release begins just in front of the replication fork and is facilitated by chromatin remodeling complexes that move with the fork. Some histone chaperones that are NAP1 and CAF1 restore the full complement of histones to daughter molecules using both parental and newly synthesized histones. Although some daughter nucleosomes contain only parental histones or only newly synthesized histones, most are hybrids of old and new. So these are sliding clamp, H2A, H2B dimer. NAP chaperone loads H2A, H2B dimer to the DNA. This replication fork goes on parental conformation, parental H3, H4 and newly synthesized H2A, H2B dimer displaced in front of replication fork. CAF1 load H3, H4 tetramer. So now we'll see DNA replication initiation in eukaryotes. So the mechanism ensures that each origin of replication is activated only once per cell cycle. An origin of replication can be used only if a pre-replicative complex forms there in G1 phase. At the beginning of S phase, specialized kinases, phosphorylates, MCM and ORC, activating the former and inactivating the latter. So a new pre-replicative complex cannot form at the origin until the cell progresses to the next step. So this is origin recognition complex. CDC6 binds with the origin, then CDT1 will bound with the MCM helicase, and then MCM helicase will be loaded on DNA. Phosphorylation of MCM and ORC occurs, and after that helicase activated, ORC will be displaced and the replication will be started. Now at the end we'll see how the telomeres replicate. Telomeres gets replicated. So there is an enzyme called telomerase. It replicates the end of chromosomes. So this is the structure of a portion of telomerase. Telomerase is a large protein RNA complex. This is telomerase protein and remainder of telomerase RNA. The RNA contains a template sequence for synthesizing new DNA telomere repeats. The synthesis reaction itself is carried out by the reverse transcriptase domain of the protein, shown in green. So what happens? This is the newly synthesized telomere DNA. And this RNA will be act as a template, and the DNA will be synthesized. This is the protein of telomerase protein. Here this acts as a palm and this acts as a thumb, like that. So how the telomere is replicating? Here you can see this is parent strand, parent strand. These are incomplete, newly synthesized lagging strand. Telomerase binds, and then direction of telomerase synthesis. This is telomerase with bound RNA template. What happens? It will increase the length of telomere. So telomerase extends 3' end RNA templated DNA synthesis. Completion of lagging strand by DNA polymerase, DNA templated DNA synthesis. Now DNA will be replicated using this strand. Any question, anyone?
Good evening, ma'am.
Good evening.
What are the enzymes involved in replication, ma'am?
In what?
What are the enzymes involving in DNA replication, ma'am?
Yes. So I will cover that. DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. So what does DNA polymerase 3 does? It will help in synthesizing the leading strand and lagging strand. Okay. And DNA polymerase 1, I told you, it will help in recognizing if any it will act as an exonuclease. Okay, I will just go into that slide. Yeah, so DNA polymerase 1 has the 3' to, like it has activity, exonuclease activity, so it can remove that, and then the DNA polymerase will proceed. So these are the enzymes important for DNA replication. Also other like DNA ligase, when we need to seal two end of DNA, we need DNA ligase. And other enzymes, if you consider, then to remove those primer, we need RNase H. This is also a DNA repair enzyme.
Thank you, ma'am.
Thank you.
Thank you, ma'am. Ma'am?
Yes, tell me.
And endonuclease also, ma'am.
What happens?
Exonuclease and endonuclease also involved, ma'am, for replication.
Yeah. To cut if there is some like wrong nucleotides added, you have to cut, right? So for that they choose off, and then again polymerization happens. So for that purpose, I will include a slide of those enzymes next class. Okay, that will be helpful, I think. Stating the function of DNA polymerases. Yeah, tell me.
Ma'am, can you share PPT to all of us?
Yeah, I have shared PPT already. You have not received it?
Yes, ma'am.
I have shared. PPT is
Okay.
yeah. So I have shared to the NPTEL committee. So I think they will share with you because I don't have direct access to share it with you guys. So that's the reason. All the lectures you can again go and visit in my YouTube channel.
What is your YouTube channel link?
Yeah, I'm just sharing. Okay.
Okay.
Just a second.
मैम
Yes, tell me.
मैम मेरा 3 वो असाइनमेंट नहीं जमा कर पाया, तो उसके लिए क्या करना होगा फिर?
Acha. Okay, okay. I will talk to the coordinator of NPTEL this course, okay? And I will let you know. What
Ma'am,
I
तो
this is my first class actually. So I also did not send anything.
माय हेल्थ प्रॉब्लम इशू, इस वजह से हमने वो नहीं कर पाया था सबमिटेड। मोबाइल मेरे पास नहीं रह पा रहा था, मैं हॉस्पिटल में भर्ती था मैम, तो इस वजह से
okay.
ऐसे ऐसे
Can
यूनिवर्सिटी में मैंने सबमिट कर दिया है, मगर अब एनटीपीटीएल का मेरे को नहीं पता इसमें कैसे क्या करना होता है।
अच्छा तो आप ये कोर्स आप रजिस्टर करोगे सर्टिफिकेट के लिए?
वो तो कर चुका हूँ मैम।
ओके ओके। तो क्या
वो तो
कर
मैं एग्जाम का भी रजिस्ट्रेशन कर दिया है मैम।
एग्ज़ाम के लिए? अच्छा तो आपका अगर आप करना भूल गए तो ना मतलब they will deduct those marks actually. There is 3 assignments are already dates are expired.
यस
So in that case you just keep on answering other week without fail, and also if there is any mail ID for communicating you can mail them, and also I will mail also to know. I don't know whether they have any other opportunities for due to medical issues. I don't know, but you can also, there is a discussion forum, you can always raise this kind of questions. They will be able to answer correctly. mm, okay, and so just for in case I am sharing one video link in this chat, you can go to that video link, and that is the channel NPTEL. The channel name is NPTEL Cell Biology. So in where
मैम
the
यूट्यूब पर।
YouTube, हाँ. So
मैम मेरे पास आता है, यूट्यूब पर मिलता है
oh
एनटीपीटीएल।
हाँ हाँ, तो मैं आई एम अपलोडिंग देयर ओनली. So
ओके।
you can if you want to revisit you can go through. Are you attending other TA's course also? So in this course there are 3 TA's. So we will be covering some topics and also assignment questions and answer, and all will interact with you. If you just now you have told some problem, will try to deliver and get it fixed, or somehow we'll get to know whether they will give chance or not. As far as I know they don't give, I think, but we can again ask. So this is all I wanted to tell you.
Ma'am, this is my first class actually.
Okay.
And I also not submitted any assignment yet
Okay.
because of gate exam, ma'am. So I am not aware of it actually.
Okay, someone has told that one of 8 assignments, 6 assignment will be count. Average of 6 assignments mark will count. Okay, okay. I was not aware of this thing because it was, I don't know, it was not conveyed to us. But it is great
Ma'am,
then. If
ma'am, it is written on that website.
oh
If
okay.
you will open that, yeah.
Okay, then it's fine. Then 2 you can they can consider, but for Abhilasha, one, I think one they won't do anything. You just concentrate on other assignments, do it well, and then there will be final exams. I think the marks are not that much. The amount of marks is not much in on assignments, but in exams also I think 70/30 or something. I am not aware. If anyone knows, you can tell. Yeah, speak it loud or tell, message here to everyone. So this is how it works. We so already, as there is some deadlines are there, so I think they won't allow, uh, but for health issue we need to check with the NPTEL coordinators.
Okay, ma'am.
Yeah, So Abhilasha, for you, I think two you were getting. You cannot submit two also, that as per as Shama, but you should now do the other weeks carefully so that you can make a good score.
Okay, ma'am. I have registered late actually, 6th of February. So,
Oh, okay.
yes.
Yeah, it's fine. Don't worry. Just you will have one final exam. Do well. That will be good. Don't worry.
Okay, thank you, ma'am.
Thank you so much. Any query so that I can raise that query in next class, as well as if we can get into today's class. I think someone has asked in chat section what is the role of telomerase. I can go over it. So we know that there is an end in the DNA called telomere, okay? But that also we need to replicate, else what will happen? That we will lose the end of DNA again and again, and the DNA length will be shorter. So telomerase is an enzyme, consists of one protein domain and RNA domain, and it will help in the replication of the telomere. Not it will help in replication. How it will increase the length of the end of DNA, so that using that strand, one RNA primer can come and bind. DNA primers can come and bind and form a primer, and eventually does a synthesis in the lagging strand. So this is the function of telomerase. It replicates the end of chromosome, and this is how the mechanism happens. I think it's clear to everyone.
Yes, ma'am. It's clear.
Thank you. Okay, so in next class I have already uploaded this PPTs in Google Drive. I have shared these links to NPTEL coordinator. Also in next class I will share the link with you in the class, so that you can also get access directly.
Ma'am, what is the basis of assignment, total assignment? Can you
If
please
I
confirm?
actually I'm not sure. If anyone knows, can you tell what is the weightage of assignment? I'm not sure. What I heard before, it is 30-70 like that, because I think there is 10 questions for 8 assignments, and there will be one exam. So I think 30 70 like that, but if anyone is sure, you can please tell, or in next class I will tell, because in the website I could not find weightage. Yeah, in next in website we, I don't know, and we were not conveyed about the weightage, but I think not more than 40, 30 70 or 40 60.
Yes, ma'am. Okay, ma'am.
Also, there is a discussion forum in the website. You can always raise your question, type your question over there. uh, TAs will be answering those questions regularly.
Ma'am, how many assignments completed yet, submitted?
3, and 4 5 are already uploaded. You can write the assignments and submit, because there is deadline is not yet come.
Okay, ma'am. Ma'am, who is joined late? So, what is the option for them?
Options.
Because the end date is 6th of February. So, I registered 6th of February only.
Okay, okay, then you could not get time, I guess, to look for
Yes,
the deadline.
yes, ma'am.
Okay, sure, I will just ask the NPTEL coordinator, and will let you know
Okay,
as
ma'am.
early as possible. Yeah,
Okay,
but
ma'am. Thank you, ma'am. It's possible, please, ma'am.
yeah,
Uh,
by the time you just finish other assignment and submit.
okay, ma'am. Thank you so much, ma'am.
Yeah, welcome. Yeah, so one person has sent that an average assignment score is 25% of average of best 6 assignments out of the total 8 assignments. So for everyone, thank you, Kiruthi, for sending this. So
Thank you, ma'am.
yeah, so thank you for sending this. So among 8 assignments, you have complete 6 assignments, and on this 21% average is there. Yeah, I knew somehow, I was remembering 30 70, but not 30 70, it's 25 75. Exam score 75 out of 100. Final score, average assignment score plus exam score. So don't worry, the marks will be get averaged. You will be eligible for a certificate only if the average assignment score greater than 10 by 25. Okay, and exam course 30 by 75. If one of the two criteria is not met, you will not get the certificate, even if the final score is 40 by 100. Okay.
मैम इसका सर्टिफिकेट अलग से मिलेगा या कॉलेज यूनिवर्सिटी मतलब जो हम लोग कर रहे हैं?
नहीं नहीं, अलग से NPTEL वेबसाइट से मेल के थ्रू मिलेगा,
ओके,
अलग से, हाँ.
थैंक यू.
If anyone want to have discussion on any topic which has been covered in previous classes, or today's classes or ongoing classes, please let me know. I will be updated so that I can deliver you in next class. If any area is not completely clear, please let us know, or I will be presenting, or I will be delivering some slides, and will interact with you in next class on week 4 and also some assignments which from the previous year. Okay, then bye everyone.
Yes,
We
ma'am.
are
Bye, ma'am. Good night, ma'am.
good night.
Bye, ma'am. Good night, ma'am.
Thank you.
Good night, ma'am.